Rồi.
Hello everyone, welcome to our class today, YB47. Today our group, under the instruction of lecturer Miss Thảo Mai, will talk about a topic of biochemistry. It's the generalization, regeneration, and synthesis of purine and pyrimidine. Next please. So you can see there is three parts of a nucleotide in the figure. A ribose or 5-carbon sugar in the middle and a nitrogen base with a phosphate group. Next, please. So before we get started with the generalization, we will take a look over a video so I hope you can understand a little bit about it.
Cells possess an enormous amount of nucleic acid. A human cell, for example, possesses almost 6 feet of DNA. So thus, nucleic acids are organic molecules which can be used for energy. And the pancreas produces both ribonuclease, which separates RNA molecules into individual nucleotides, and deoxyribonuclease, which separates DNA chains into individual nucleotides. In the small intestine, brush border enzymes include nucleosidases and phosphatases. These break apart the parts of a nucleotide so that the nitrogenous base, pentose sugar, and phosphate are separated. And these components can be then absorbed into intestinal cells through active transport.
So from that video, I hope you will understand a little bit of the generalization. To talk more about it, nucleic acids are polymers of nucleotides. The latter are composed of a 5-carbon sugar, it can be ribose or deoxyribose, linked both to a nitrogen base. It can be purine or pyrimidine and a phosphate group. The purine and pyrimidine nucleotides, which occur most frequently in the nucleic acids of cells, in the unpolymerized stage, they occur at nucleoside diphosphate (NDP) and triphosphate (NTP) as well as the monophosphate forms illustrated. In the polymerized stage, the phosphate on the 5' OH of the sugar is linked to the 3' OH of the extraction sugar residue, so that nucleic acids consist of a backbone of alternating residues of a sugar and a phosphate with the base projecting out from this. Two types of nucleic acid is deoxyribonucleic acid, also called DNA, is a polymer of deoxyribonucleotides, and ribonucleic acid, RNA, which is a polymer of ribonucleotides. Nucleic acid metabolism is concerned with the synthesis and breakdown of purine and pyrimidine nucleotides. The utilization in DNA and RNA synthesis and the structure, localization, and function of these nucleic acids. Nucleic acids are the most important organized macromolecules carrying, preserving, and communicating genetic information. Allows organisms to transfer genetic information from one generation to another generation. These macromolecules store genetic information that identifies traits. Next, please. So we will move to digestion. As you can see, the nuclease, um, or it is known as a phosphodiesterase enzyme, belong to the hydrolysis enzyme, have to segment the linkage of phosphodiester of DNA into mononucleotide and nucleotide. There are many types of this enzyme. For example, it can segment one strand or two strands of DNA or only RNA chain. And this process only needs energy to for this enzyme to fulfill its function, and the energy source is ATP. The next enzyme you can see is nucleotidase from intestine is a phosphatase enzyme. Have to hydrolyze nucleotide into nucleoside and phosphoric acid. Um, Next, please. About the absorption, we have the phosphate um, is added into phosphoro reaction, and then it can be and the nucleoside then is hydrolyzed into nitrogen base and pentose with the help of nucleosidase enzyme. Then the pentose continue um, on the metabolism of glucose. Nitrogen base is break down. Finally, we can have the products. Is the products are uric acid, urea, and CO2. The nucleosidase also plays a role like an enzyme for the phosphorus reaction. Next, please. So this is the summary diagram of nucleic acid degradation. I'm sorry. So you can see this is the summary for you to have a total look about the topic. Next, please. Um, this is about the purine-based degeneration. We have two main types of reaction: hydrolyzed amine reduction and oxidation. You can take a look over the what can I call it? Like the diagram. Yeah. Next, please. And this is the end products of the purine-based degeneration. Like I talked to you before. Next, please. Well, there are also some unfortunate or unwanted disease caused by disorders of purine degeneration. For example, here we have, I'm sorry, can you turn back on the slide? Yeah. Um, as you can see from the uric acid, we have the hypertension or neurodegenerative disease or gout. Another example, for example, like the CN2, we have the spastic paraplegia 45 or cancer. So next, please. This is like the popular disease caused by the abnormal degeneration of purine. This is gout disease. The normal uric acid in blood um, comprise of 3 to 6 milligram percent, while in abnormal body that suffer from gout disease, the uric acid in blood consists of more than 10 milligram percent, even to 15 or 20 milligram percent. This is due to the increased blood uric acid and this can lead to sodium urate, so crystal less soluble, or stagnation in small chunks, for example, big toes and tablets and champagne. Next, please. As you can see, this is the figure about gold disease. We have the size and the mechanism. Next, please. So, fortunately, we have the treatment for this disease. It is the name of the medicine is allopurinol. So this medicine will help to inhibit the enzyme statin oxidase in the treatment of gout. So that is my part, and we will move next to the representation of my friend Thiện Phú.
Okay, hello everyone. I'm Thiện Phú, and now let's move to the first part of my talk, which is about kidney stones in degeneration. In the kidney causes kidney stones, urinary tract stones, we have two reasons. First, increased blood uric acid synthesis, and second, decreased renal excretions of uric acid. Next, please. And the second part of degeneration is degradation of pyrimidines. The cytosolic pyrimidine nucleotide is degraded to the bases uracil and thymine. Uracil and thymine are further metabolized in the liver through a reduction reaction. Next, please. Uracil and thymine are the, the end products of pyrimidine catabolism. In addition to NH3, CO2 are alanine and amino isobutyrate, which continue to be metabolized as other amino acids. Next, please. Then they form malonyl CoA by precursor for fatty acid synthesis, and methylmalonyl CoA by converted to succinyl CoA to enter the citric and cycle. And next, please. Next, please. Finally, I will go on part 3 of our topic, synthesis. Next, please. First, synthesis of purine and purine nucleotides. The purine skeleton is a molecular structure that includes in 6-member pyrimidine ring fused to a 5-member imidazole ring. Purine biosynthesis occurs in the cytosols of all cells. This process consists of 3 main stages: PRPP synthesis, IMB synthesis, and AMB-GMB formation. Next, please. The first step in purine synthesis is the formation of PRPP from the ribose phosphate sugar catalyzed by an enzyme called ribose phosphate pyrophosphate. PRPP has a very important role in both the synthesis of nucleotides and some amino acids. PRPP is immediately synthesized as a feedstock for an 11-step reaction, and the final product of this reaction is inosine monophosphate. IMB is the precursor for the synthesis of two types of purine, AMP and GMP. Next, please. Okay, the PRPP metabolisms were analyzed as follow 11 steps. And next, please. Through 11 transformation steps, we can identify the origins of the atoms in the IMB molecule as follows. First, glycine and carbon atom at position C4, C5, and C7. And N10 forming tetrahydrofolate as a carbon atom at the C2 and C8 positions. And then aspartate as a nitrogen atom at the N1 positions, and glutamine as a nitrogen atom at the N3 and N9 positions. And nextly, the third stage of purine synthesis is the formations of AMP and GMP. First, the formation of AMP occurs through two steps. Step 1, formation of the bond between the amino acids aspartate and AMP. Step 2, the enzyme adenosine catalyzes the removal of the fumarate moiety from the adenosine molecule, thereby forming the molecule AMP. And next, please. The formations of GMP also occur through steps. And step 1, the hydrogenation of molecule IMP, and step 2, the mediation of the SMB molecule to GMP. And next, please. We move to part 2 of synthesis of pyrimidine and pyrimidine nucleotides. Since pyrimidine molecule are simple tambourines, so is their synthesis simple but is still from readily available components. Glutamines, amide, nitrogen, and carbon dioxide provide atoms through and 3 are the pyrimidine ring. They do so, however, after first being converted to carbamine phosphate. The other four atoms of the ring are supplied by aspartate. As is true with purine nucleotides, the sugar phosphate portion of the molecule is supplied by PRPP. And next, please. So we have two stages of synthesis of pyrimidine and pyrimidine nucleotides, and stage 1, formations of uric acid. Uh, next, next, please. Before the formations of uric acid take place, pyrimidine synthesis begins with carbamine phosphate synthesized in the cytosols of the tissue capable of making pyrimidines, highest in spleen, thymus, GI tract, and testes. This uses a different enzyme than the one involved in urea synthesis. Carbamine phosphate synthesized through refers glutamine to free amino and has no requirement for an acetyl glutamate. And next, please. Carbamine phosphate condenses with aspartate in the presence of aspartate transcarbamylase to yield and carbon aspartate, which is then converted to dihydroorotate. In man, CBS through transcarbamylase and dehydroorotate activities are part of a multifunctional routine. Oxidation of the ring pire complex, poorly understood enzyme produces the free pyrimidine uric acid. This enzyme is located on the outer face of the inner mitochondrial membrane, in contrast to the other enzymes which are cytosolic. Note the contrast with purine synthesis, in which a nucleotide is formed first, while pyrimidines are first synthesized as free base. And next, please. Finally, I will go on to talk about stage 2, formation of the nucleotides. Uric acid is converted to its nucleotide with PRPP. OMP is then converted sequentially, not in a branch pathway, to the other pyrimidine nucleotides. The carboxylations of OMP gives UMP. OPRT and OMP decarboxylase are also multifunctional protein. After conversions of UMP to the 3-sulfate, the amide of glutamine is added at the expense of ATP to yield CTP. And synthesis through regulations, the controls of pyrimidine nucleotide synthesis in man is exerted primarily at the level cytoplasmic CBS2. UTP inhibits the enzyme competitively with ATP. PRPP activities, other secondary sites of control also exist. That's all.
So thanks very much for paying attention to the presentation of our group, me, Minfei, and my friend Hing Fu, and our group. Thank you very much.
Thank you.